Hello, welcome to 13 Universal. This is A-level CIE Biology, and we want to continue to solve questions on planning, analysis, and evaluation, paper 5, series 4. This is for done 1, 2, and 3. So get your writing materials and let's practice. Remember the drills. When I'm done reading the questions out, you pause the video and solve the question by yourself. Please do not check the math scheme. Do not go to solution without solving the question. All right? That's how we roll it. So this is the first question. All right? We have a whole lot of them, and of course, please make sure you flow with the ground information, the background information. Very important. Okay? As you read critically. Some students wanted to investigate the effect of light intensity on the rate of photosynthesis in Brazilian waterweed, Egeria densa. This plant is found in freshwater ponds. The students use apparatus shown in Fig 1.1, right, in their investigation. A strand and a clamp were used to hold apparatus in a vertical position. Okay, that's it there. Yeah, before you go on, guys, remember, like I told you, read critically. Look at the statement. Some students wanted to investigate the effect of light intensity on the rate of photosynthesis. So the first question I want to ask you is, all right, which one is the independent variable here? Look at this is effect of what? Light intensity. Is it light intensity or rate of photosynthesis? Which one is the independent variable? You have to identify it as you read. ID your independent variable and dependent variable. So your independent variable is the one you are changing. Your dependent is the one you are going to measure. All right. So which one are you going to change? Which one are you going to measure? Okay, are you going to measure this? Of course, this is your independent variable. You're going to be changing this and you're going to be measuring rate of photosynthesis. Okay, so this affects this. Independent variable affects dependent, right? Dependent is measured, independent is altered or varied. Don't forget, guys, that this is very important, that independent variable, all right, is measured, and there are other variables that we also have to keep constant, right? So you have to look at this diagram. It will show you how your independent variable is changed, and how your dependent variable is going to be measured. How do we measure rate of photosynthesis? Okay, it can be measured in the, look at it, see. Can you see? This is a plant, water. It's called what is the name of this? Egeria denia. That's the, okay. So during photosynthesis, it produces air bubble, which is oxygen. All right. So this is likely going to push exact pressure here, which will push the meniscus to move. So most likely, the rate of photosynthesis will be measured, all right, by measuring the distance moved, okay, by the meniscus along the scale. Okay, so you see, you're going to measure distance moved, all right, by the meniscus along the scales. So there are things you must keep constant there. You must keep the species, the length of this plant, the mass. Now look at this carbon hydrogen carbonate ion, which is the source of carbon dioxide, must be kept constant. This must be kept constant. The concentration must be kept constant. The volume must be kept constant. All right. These are things you're going to note before you go into the question. So identify your independent variable, how to alter it, the dependent, how to measure it, and then your standardized variables, which ones are going to keep constant. So we've identified, all right, a couple of them here. Okay, let's go to the question. It says suggest suitable control for this investigation. All right, now the question I'm going to ask you as you think about it, what is control? What is control? But okay, before we go to that question, let's read this. The student carried out these steps to set up the apparatus shown in Fig 1.1. The secret to paper 5 is that they will always give you clue. The clue will help you to design the experiment they will ask you. So some sodium hydrogen carbonate was added to a sample of pond water as a source of carbon dioxide. So that's the source. The syringe barrel was filled with pond water containing sodium hydrogen carbonate. A fresh piece of E denser was cut underwater. the cut stem was quickly placed in the pond water in the syringe barrel. So these are the sequence of events. The plunger was replaced as shown in figure 1.1 and adjusted until the meniscus in the capillary tubing was near the top of the scale. A bench clamp was used as a light source, so we use a bench clamp, a bench lamp light source. So one, okay. The students noticed that bubbles of gas were given off from E denser as the gas collected in syringe barrel. The meniscus, you see, they're giving a clue, moved down the capillary tube. The plunger of the syringe did not move, but the meniscus moved because of the gas exerting pressure. The student used the apparatus to investigate the effect of different light intensity on the rate of photosynthesis in denser. Okay, let's get back to this question. Pause this video and solve this. It says suggest a suitable control for this investigation. Suggest it. Pause the video and try to solve it. Okay, now control experiment is that setup that shows us what happens if the independent variable is removed. Okay, now independent variable here is light intensity. So if we remove light intensity in the dark, so let's find out whether it is light intensity that is producing the effect. Is there any other factor that is causing the change? All right. Now, a control also means you're taking out, all right, the organism or the factor, the substance that is causing the change. It may not be directly the independent variable, maybe the organism or the factor that is causing the change, apart from independent variable. Okay, independent variable is the light intensity we're changing, but the factor that's causing the change also, apart from independent variable, is this plant. The plant is carrying out the photosynthesis. If you remove it, have a setup where it is not there, that's your independent variable. If you carry out this experiment in the dark where light is not there, removing light completely, okay, that is also your independent, your control variable. You are taking away something that is causing the change, okay? But in this case, you can choose taking away the plant, okay? All right, because photosynthesis also does occur in the dark, like independent, all right? So taking away the plant, all right, helps us to know that without that plant, it will not occur, all right? Just one mark, look at it. Idea of replace water plant. Take it away and then replace it with what? Dead one, okay? Of the same volume. If you don't have it, you can put inert, all right, or glass beads, all right? Or or bond water with sodium hydrogen carbonate. No water. Take away the water plant or replace it with something else. Okay. So look at the next question. Describe a method. Yeah, they will always ask this question. This is 7 marks question, right? Describe a method using the apparatus shown in figure 1.1 that a student could use to investigate the effect of light intensity on the rate of photosynthesis of E denser. Your method should be set up in a logical order and be detailed enough to allow another person to follow it. The steps carried out to set up the apparatus shown in figure 1.1 should not be included, guys. Remember what I told you in the other videos. Anytime you are dealing with method, but I want you to pause this video and do this method by yourself. Let's see what you got. Okay, try to pause it. But the first thing, because it's a structured question, you have to respond an essay, an extended, all right, question, 7 marks. You have to write a prose, a descriptive essay of what is happening here. But before you start writing your essay, you need to have an outline. Okay, please make sure you have an outline before you go into it, so that you don't just jump into the question and you run out of ideas. Biology, all right, has an essay side of it, and you use the same skills that you acquired in English language while writing your essays. All right, you must have an outline. I want to write an outline there. So that's an outline. So what is your outline here? So in your outline, what should be your outline? So you get your outline ready before you start writing. Okay, so in my outline, first of all, you must identify your independent variable. Okay, we have identified it, and then you must also not just identify it, you must state how to vary it, how to change it. How do we change our independent variable? You state it or use your imagination. Okay, and your lab skills from paper 2. Okay, for example, your independent variable here is light intensity. All right, how do you vary light intensity, guys? Okay, so what you're going to do, because they gave us a clue, you use a lamp, okay, a bench lamp. So if you have a setup, okay, so because we have only one lamp, okay, let's assume that this is our setup, like the one they drew there. That is what is it called, that diagram there, the setup, okay, the setup, having the plant and all of that inside, okay, you know that, so don't bother about my diagram, okay. So you have the plant inside here, okay, yeah, the plant is inside here, and you know that this is a tubing connected to a scale, just to show you, um, okay, this is connected to a scale, and there's a meniscus here. Now you can check the real diagram there, and there's a scale, okay, yeah, okay, I'm just trying to, yeah, so this is the lamp bulb, the lamps, okay, bench lamp. Now, because we have only one of one light, okay, light intensity. So how do you vary the light intensity? Okay, what you do is that you will now vary the distance from the lamp. Okay, this is distance from the lamp. Sorry, distance, yeah, of the lamp from the setup. So that's distance, okay, at intervals. So you put it here, you can decide to vary it, all right, another distance from here to here. So remember, so this is the light, okay, the lamp, the bench lamp. You can start with that first distance and adjust it, right, to the next distance, right. So as you are taking it away from the setup, what is happening to light intensity? Is it increasing or decreasing? What do you say? Is it increasing or decreasing? Of course, it is actually decreasing the farther away it is from the setup, but that's one way we can vary it. All right, okay, so this continues. All right, so the distance from here, all right, to set up to the next one. So you remember that your independent variable, you have to get a range of value. So I can decide to say, okay, I'm going to you know, the range should be from 10, okay, of course, maybe 50 to 0. All right, 50 to 0, and then, you know, between 50 and 0. Is that okay? I'm going to get values from that. Okay, it's 10 cm, okay, 10 cm cube, 20 cm cube, that's okay. I'm getting, okay, four other values, 30 cm, sorry, not cube, just 10 cm because distance, 20 cm, 30 cm, 40 cm, and then you have 50 cm. Is that okay? Yeah, 50 cm. So I'm going to state it here, the distance. So I'll start with the first distance, which is 10 cm from, all right, I put the light source 10 cm from the experimental setup, okay? All right. So of course you're going to state that you fill in the barrel with hydrogen, okay, sodium hydrogen carbonate solution, and then put the same mass coming to that. But you know that you're going to put in all of this, state how you're going to do that. But before you start writing, you must know what you're going to write. So I'm going to put this as my independent variable, the values, okay? Now, having said that, the next thing I'm going to consider before I start writing is I consider my dependent variable. So, dependent variable, okay? My dependent variable. This is independent variable, IV, sorry, that's IV, independent variable. So my dependent variable is rate of photosynthesis. How do I measure it? Okay, how do I measure it? And you know, they have given you clues. So dependent variable is the one you measure. Okay, so how do you measure it? You must find that before you start writing. From the diagram, from the information, the background information given to you. So how do you measure it? First of all, you have to ID it, and okay. So how are you going to measure? You're going to measure the distance moved by the meniscus, right? The distance moved by the meniscus. That's how to measure the rate of photosynthesis. The distance moved by meniscus. Okay, if the distance is longer, that means rate of photosynthesis is longer. So it's high. It's high. If it's shorter, the rate of photosynthesis is low. Okay. So then of course you have to also be able to identify your key variables. What am I going to standardize? This I'm preparing my outline. Key variables and my key variables here, all right, include right, the same species of water plant, the same mass, okay, the same length, and of course the hydrogen hydrogen sodium hydrogen carbonate concentration should be the same, all right, the same concentration, okay. So the time in before light is should be the same, all right. Okay, now of course I have the same light. Now if I have different lamps, that's fine. If I have different lamps, I'll just put it in one distance and keep changing. So but I have to determine the power rating, okay, of the different lamps, okay. But if I have only one, I adjust the distance. If I have many lamps, I use them, put them in one distance. Distance become constant. Now, but look at this. My you have to put this right constant. Your key variables should is the one that you put. You keep constant. I've mentioned a few of them. Okay, now I'm preparing my distance. All right, and then of course you have to talk about hazard, validity. How do you make it valid, right? By repetition, right? You are going to talk about how to make a result valid, repetition, and how you're going to talk about hazard, risk, and of course precaution. Okay, you are dealing with sodium hydrogen carbonate is a chemical. Is it an irritant? If an irritant or corrosive, okay, what would be my precaution? I wear gloves. Do you get it? All right, this is very important. So guys, these are the most important things that you're going to write in your method. Okay, but in now start writing, you develop the essay. Okay, so you develop the essay, right, by stating, for example, how you know this is prepared. You pour same volume, same concentration of sodium hydrogen carbonate into this barrel, okay, of the syringe, and then put the plant. The plant should have the same length, same get species, and cover it, put the plunger here, okay, and then set up your light and allow it to acclimatize. Then after some time, you start taking the reading moved by the meniscus for each of these, all right, distances. So they are placed at different distances, okay. So you just mention that your constant variable mentioned it, and then for each of these distances, you have to take, replicate value 3, repeat, and then calculate your mean, all right? And then for hazard, you mentioned some of them that are hazard here, were mentioned it, all right? Now these plants can even be an allergen, all right? So you wear nose mask, is that okay? All right, let's see what the mask says. Look at it. They're always asking this question, guys. Let's get this right. So you see, number one, use lamp at different distances, like we said here. Okay, but we're going to put this in an essay form, right? Use lamp with different power ratings. If you don't want, if you have different lamps, but if you have one, vary distance from the experimental setup. Okay. Number two, use minimum 5 different stated light intensities. Okay, here we're using distance. Okay, but you can convert the distance also to light intensity. So you see, light intensity can be converted to distance. I think there's a formula for that. Okay, I think that formula is 1 all over the square of distance, right, from the setup, right? Yeah. Okay. Yeah, because there's a inverse proportion in relation to them. So as you move it away, light intensity decreases. Okay, so look at it. So use light meter to measure light intensity. Okay, and of course carry out investigation in the dark room for your control. Measure the distance moved by the meniscus, or measure record distance moved. Okay, stated length of water, please attempt to keep constant. This is a simple thing to do, guys. Okay, now of course you have to reduce temperature, all right, the heating effect of this lamp. Okay, not really, you just put so it absorbs heat because the lamp produces heat and heat, all right, introduces temperature. So temperature can actually affect the rate of photosynthesis. So you have to remove it by absorbing, reducing the heat effect, right? All right. Okay, so the idea of equilibrate acclimatization of this in the light before you start measuring the distance moved. Okay. Okay. So let's go calculate the mean, set out. Okay. 7 marks. Quickly. Complete the sketch graph to predict the result that you would expect from method you have given. All right. So what is the relationship between light intensity and rate of photosynthesis? Here, of course, if we are measuring, of course, this is it is light intensity. But here, if you use distance, this is distance d in centimeters. All right, that is my independent variable. That's why I adjusted. I varied my this thing, the light intensity, right? And of course, we have what is it called? This is also okay. That's your light intensity. Okay, you know this is representing your light intensity. Okay, then you put the unit. If you don't want to put that, put, of course, you need to convert the distance to light intensity. The unit, all right, put the unit here, which is candela, all right, and then you put distance, all right, moved by the meniscus in centimeters. So in order to prevent confusion, right, remove this and use light intensity. Convert the different distances into light intensity and put the units for this. And of course, you know that as light intensity increases, rate of photosynthesis will increase. Okay, so rate of photosynthesis will increase and the distance move will be longer. So it will go, there's a positive correlation, reaches the peak and then levels off. At this point, light intensity is no longer the limiting factor. Okay, so this is how it should go. This is light intensity because that's an independent variable. This is rate of photosynthesis, but we're using distance moved by meniscus. So the higher the light intensity, the more distance moved. Okay, until reach the plateaus and levels. So that's all you need. Put the unit and remember that independent variable is always on x-axis, dependent is always on y-axis. Okay, and then you predict based on your theoretical knowledge of this. Okay, look at it, guys. It's a horizontal axis level distance from light bulb. You can use distance from light bulb or light intensity or power ratings. Any of them can here. Vertical axis, that's independent rate of photosynthesis, can convert to rate or rate of movement of meniscus or distance moved. Meniscus moved, any of them can be there. And then set time taken for meniscus to move. You can use time taken for meniscus to move. All right, a set distance. And then distance from light bulb, these are correct unit. You must put a unit distance from light bulb, cm or millimeters, light intensity lux or candela. Alternative power ratings, you see that watt. Okay, rate of photosynthesis, you can use centimeter or in minutes. So whatever the distance moved divided by the time taken to move that distance. Okay, so you can see that. All right, and then what else you do? See the shape, positive gradient may level off at high intensities. So positive gradient levels off at high intensities. Okay, negative gradient may level off. Okay, high intensities. That is if you are doing, of course, distance against photosynthesis. So see, as distance increases, photosynthesis will decrease. But if you're using light intensity as increase, it will increase, so a positive correlation. Negative gradient may level off. Okay, so when it gets, all right, somewhere here, it levels off. If we're using distance move, a distance of the setup, okay, against rate of photosynthesis. All right, remember that as distance increases from setup, this is going to decrease. All right, okay. But if you're using light intensity, it's going to increase. You get? But when it reaches a particular point, it will still level off. Positive gradient may level off at shorter distances for sure. Okay, distance from Bob. All right, ask us your question. That's 3 marks. Yeah, the student decided to use paper chromatography to separate the photosynthetic pigment found in E. densa leaves. Okay, E. densa leaves were ground up in a small volume of solvent to make a concentration solution of pigment. The different pigments in the solution were separated using paper chromatography. The distance moved by the solvent was measured on the complete chromatogram. The distance moved by the pigment were also measured. The area values were calculated for these pigments. Okay, this table it's a complete table 1.1 by calculating the area value for pigment 2. Give your answer to two significant figures. The formula for the calculation RF is there. There's a formula there. Okay, so let's get it sorted. Yeah, so I want you to pause the video and try to do the calculation. Pause the video and try this out. Okay, so please try this out. Pause the video and try it out. Okay, yeah, so pause it. So they have given us a formula. So now the distance moved by the pigment. Okay, yeah. So if you come here now, see the distance moved by solvent, so move the pigment is 94. Okay, so just put 94 here, guys. It's simple. That's distance moved by the pigment. And then divide it by the distance moved by, because over distance moved by solvent, which is 107. 107. Okay, so this will be all over 107. 107. So punch your calculator. What does it give you? So punch yours. Let me punch mine. So 94 by 107 gives me, now they said in two significant figure, right? So what does that mean? So it will give me 0. So to convert it to significant figure, 0.8. Okay, 0.8. Okay, according to what I have here, 88, right? 0.88. What do you have as area value? Okay, 0.88. All right, let's go. 1 mark. Now they said the students use a published material considered. Use the published source to find area values for 3 components that they're expected to be present. This is the published one, so we use this to compare with what we worked out in the experiment. The student correctly identified pigment 1 as carotene, pigment 3 as chlorophyll A pigment by comparing the values from the table and this. Okay, that's how you, as chlorophyll B, and the calculated area values were not exactly the same. They were not exactly the same, so there were some kind of errors as the error values in the published source. There's always experimental errors. Okay, kind of errors. So let's see. Describe one difficulty with paper chromatography that could explain why the error values calculated by the students were not exactly the same as the published data. Okay, now it is difficult. Remember that during your chromatography, guys, all right, you need to have your chromatogram, which is your paper, right? This is your paper here. Okay, yeah, paper here, and then you draw your baseline using pencil. Then you use pencil, and then you're going to dip it into the right, okay, into the right solvent. But before you dip it, there's something you do before you dip it. Of course, you draw your baseline. Okay, this is your solvent, and then you have to put the extract from the leaf. You have to concentrate. You make a concentrated, okay, spot. Now it's difficult to make this concentrated spot because you have to add, let it dry, add again, let it dry. You need to be concentrated. Okay, so it's difficult concentrating or making a concentrated spot at the baseline. Okay, now guys, another problem challenge is that sometimes because once you dip this, okay, the tip of this paper in the relevant, okay, suitable solvent, this will move up. Okay, this is the mobile phase. Okay, it will move this different pigment based on their solubility will start moving up. Of course, the solvent will always move to the end here. We call it solvent front. Okay, yeah. So but these other ones will start moving. So So sometimes when they move, they overlap. This solvent A overlaps with solvent B, sorry, pigment A overlaps with pigment B. So they spread out. So that spreading out creates, all right, error, confusion, okay? So you see them overlapping. This is another pigment here, okay? This is another pigment. This pigment, okay, overlaps with that pigment. See this pigment, okay, overlaps with this pigment. It's difficult to now determine it overlap here. So two of them overlaps. Do you get? So it's difficult to determine that, right? So these are difficulties. And of course, guys, if you check, you find out that if you don't have a suitable solvent, it can, right, determine their solubilities and how they move. So they introduce errors. So let's see the masking. Difficulty in achieving concentrated spot of extract not spread out. Each pigment spread out during migration. They spread out, that's the problem. Give long streaks, runs into each other, overlap any of them. Please memorize it. Pigment may fade too pale to see clearly, so it's difficult to measure accurately. Okay? Because you know that if you're going to measure from here to here, divide by here to here. This is distance moved by the solvent, this is distance moved by each of the pigment. Please don't forget. Okay, enter may determine that you measure. So it's difficult to measure from here to here for this because they overlap, all right, each other. Distance move here for each of them all over. Distance move by the solvent from here to there. Okay, so it's difficult. You can see that quality, chromatography paper, difficult to get a type of solvent, right, and other ambient conditions. One mark for all of this. Yeah, welcome to this one. Is an invasive alien species is a species that has been introduced into an ecosystem where it is not normally found and causes harm to this ecosystem. Invasive alien species can change habitat. Remember, when you are reading this, read it technically. Okay, think alongside. So invasive alien species can change habitats, reduce biodiversity, see what it causes, and cause extinction of native species. Salt cedar trees, tamarisk species are invasive. These guys are invasive, okay, or alien species introduced to North America from Europe and Asia. So from Europe and Asia, introduced to North America in the early 800s. Okay, a biologist carried out an investigation in a woodland ecosystem next to Virgin River in Arizona, North America. In the woodland, there were areas that contained only salt cedar trees, areas that contained only, okay, and areas that contain a mixture of native tree species and salt seed trees. When you get to such point like this, you pay attention. So because something is changing, and when you get to a point where something is changing, that gives indication of which variable changes or varies. You're right, independent variable. So this gives me an idea. In the woodland, so some parts contains only salt cedar trees, other part contains a mixture of, all right, salt cedar trees. And all right, let's go on. The biologists wanted to test the hypothesis that the diversity of rodents or mice or rats is lower in areas with only salt cedar trees than areas with a mixture of native trees species and salt cedar trees. So that's what we want to test. Okay, yeah. So what is independent variable here and what is dependent? Of course, that's the first thing they ask you. Identify your independent variable in this investigation. So the first question you ask yourself, which one is changing? Which one are we changing? Right? Okay, so they want to measure diversity of rodents in areas with only what? So see that trees, okay? All right, they want to check for that, all right? So they're looking at areas with only salt cedar and areas with mixture of salt cedar tree species and salt cedar, mixture of native trees species and salt cedar. So what are we changing here? We're changing the presence of, all right, native, all right, native tree species and of course native and the absence. That's what we're changing. Or we're talking about woodlands, different types of woodland. One type of woodland contains salt cedar, the other one contains, okay, native tree species and salt cedar trees. So that's what we're changing. Okay, so look at it here. Type of woodland. presence or absence of net. So type of woodland is the independent variable. So that's what we're changing. One has source cedar only, one has both source cedar and native tree species. Okay, presence or absence. Okay, so look at that's one mark. All right, the biologist decided to trap rodent in an area with only source cedar trees, and to trap rodent in an area with a mixture of native trees species, okay, and source cedar trees. The biologist, now this is what the biologist did, and I want you to pay attention here. All right, place 25 small mammal traps at random sites, right, in area of woodland. 25 small mammal traps, 25 small, baited each trap with 10 grams of food for rodents. Checked each trap after 24 hours. So he did all this for each trap. What does it tell you? These factors, what are they? Are they independent or dependent or key variables? Identified any rodent caught in the trap, marked the rodent with an air tag and released them back into the woodland. Carried out this trapping process 4 days in April. Did it in April. So there's the number of different individuals of each rodent species trapped during the 4 traps was recorded. Okay? Now, the first question here, pause the video and solve it. Identify 3 variables other than the small mammal trap and ear tag, all right, that the biologists standardize in this investigation. So which one is the key variables? That's the one we standardize. So you go come here, okay. All right, remember that the number of small mammal trap. So they say we should not mention the small mammal trap. We shouldn't mention the type of it, but we can mention the number. So each woodland, all right, each site has 25. Okay, so it must be 25. They standardize it. Okay, and if you check the food they baited, 10 grams for each site where they put it. Okay, and you see the checking time 24 hours. So these are the independent, the constant variables, right? See, they call the standardized variables. 25 traps, same number of traps in each area. That's each area. 25 traps, same number of traps in each trapping season. Mass, okay, of food for rodent, the same bait, time interval for checking, before checking, okay, before checking, 24 hours for every trap, same number of days session for each area. So these are the things is standardized here. So they didn't change it, did it for every trap. All right, that's one mark for one of them. Describe a method that the biologist could have used to select the trap site randomly. Anytime you want to select, all right, the trap site randomly, pause the video and then let me know. Okay, so first of all, you're going to have to grid the place. All right, have a vertical, okay, line, horizontal line. Okay, you now use random app generator. Use random app generator to get coordinates. Okay, if you type it, it may tell you maybe from this point, maybe 0, we have about 7 cm, which may be something like it. Okay, now horizontally, maybe about 3 cm sometimes. Okay, so use them as coordinate. Trace 2 of them. Okay, yeah, they form your coordinate. Whatever they meet, but you must use random app generator to find this coordinate. You place your trap here. Okay, your trap will be here. We use the same thing for quadrant, for dropping quadrant, because in random, okay, sampling. So use also, now you can try also using the same random app generator. It can give you 2 other coordinates. Maybe one is here, okay, you trace it, okay, the vertical one, and the other one is here. You trace the number, you measure it with your line or ruler, and then drop this trap here randomly. Okay, look at it. Suitable method of generating random number. I can use an app, coordinate position, use the number generated as coordinate, grid reference, locate each trap site. Okay, that is it, guys. Table 2.1 shows the result of the investigation. That's it. All right. Simpson's index of diversity is a method of assessing biodiversity. The formula for Simpson's index of diversity, that's it, guys. That's the formula. Okay. So whatever you do here, you minus it by -1 from it. The number of each individual, small n, number of individuals of each species present in the sample, the big letter, capital letter, uppercase, total number of all individuals of all species present. So let's look at what they want us to do. Now guys, it's time to play games of the mind. Let's do this. The Simpson index of diversity D for the area with only source cedar. This is it. The one that has only source cedar. Remember, we have two areas here. So source cedar is this. Okay, that's it. Simpson's after calculation we have this, right? 0.531. That's okay. The next question says calculate a Simpson's index of diversity D for the area with a mixture of native trees and species and salt cedar trees. You may use table 2.1 for your working. Write the value for Simpson index of diversity D on the dotted line. Give your answers to three significant figures. Okay, so I want you to do this with me. Remember, this is n. Okay, this is the total number of species, 9. So the first one you do is you say 9. Okay, bring your calculator. Let's play game. Okay, so what's the first thing you do there? You say 9 for the first one. You say 4 divided by 9. Okay, what does it give you? 0.44. Okay, all right. Right, you can just pick 3, 0.44. I make it 44. Okay, now you do the same thing here. All right, you can also decide to say 0.44 squared. All right, whatever you want to do, so you just square it. Okay, so 0.44 squared. Is that okay? So that's 0.44 squared. Is that okay? But what does it give you? So I have 0.9. Okay, I have 0.19. When you square it, yeah, I hope you are doing this. 197. Okay, whatever I have, I square it. Okay, so I go to the next line. The next line is 1 divided by 9. I hope you are doing that. 1 divided by 9, that's 0.111. Okay, 0. What? 1. So I'll then square it. Okay, I'll square it. Yeah, so that gives me 0. Yeah, that gives me 0. What? 0. Yeah, 0.2. That's 0.0. Sorry, 0.0. 0.012 approximately. So I'll do the same thing here. 0.1. Okay, yeah, 1. If I like, I make it. Okay, I can put 1 here. Quite the same thing. 4 here, same thing. Okay, make it 3, and it gives me the same thing. 0. 0. That's point here. 0.012. Okay, so I do that for 2. So 1 divided by, that's 2 divided, we're done with 1 now. 2 divided, okay, so we're going to the next one. 2 divided by 9, right? 2 by 9 gives us what? Also, it gives us, okay, 0. Oh, that's 0, sorry, 0.222. Then you square it. Okay, 0.222 squared. Okay, what does it give you? 0.049. That gives you 0. Okay, 0.049. Yeah, so that's 0. This is 0.049. Sorry, we're taking time just to show you this. Right, 49. And the last one, the same thing. We have 0 point, okay, 1, 1, 1, which will give us 0 point, okay, yeah, 0, 1, 2. That is it, guys. So we're going to add up everything there. Okay, I'm going to add up everything. So I want you to get your calculator and add up everything. Okay, I mean, I could confirm. Okay, so add up everything there. So let's go. So the first one, 0 point this. Let's add up everything. 0 point this plus this plus 0 0.197. Okay, 97 plus 0 0.10, no, 0 0.0, yeah, 0 0.0, yeah, that's 0 0.012, okay, plus another 0 0.012 plus 0 0.049, okay, yeah, good, plus 0.012. So the total gives us 0.28. So that's 0.28, guys. So after adding up, so we have 0.28. So we have gotten that 0.2828. We have gotten summation of everything. 0.282. Okay. Remember, what we just did now was summation. This everything we did here now, okay, is summation of that n over capital letter N, right? Yeah, all square. That's what we did. That's 0 point. That's okay, square. Yeah. So remember that our d, all right, is 1 minus that. Okay. 1 minus 1 minus what we just calculated, which is this, everything here, 1 minus this, and this is this, okay, I follow. All right, which is 0 point, right, 282, okay, that is Simpson's inverse of diversity for the mixture of salt cedar and the native species. So guys, yeah, so you just do 1 minus it and let's get the answer. The answer will be 0, okay, yeah. So the Simpson index diversity for salt cedar and the mixture of native trees is 0.7, okay, 0.78. But they say we should write the answer, calculate the answer in what? They say we should write the answer. Wait, okay, 3 significant figure, yeah, 3 significant. So 0.7, okay, 18, okay, that is it. Can you see that this 0.718 is bigger, all right? This is bigger than this. So this shows this has more biodiversity than this, okay? All right, but let's check the math scheme here, okay? Yeah, they showed us how to do that. So we got, see what they got, okay? 0.716, whatever, okay? We got 1718. So, yeah, 2 marks. So it's time to, they say, with reference to table 2.1 and values for D in the 2 different areas, state and explain the conclusion that can be made from the results of the investigation. So the conclusion that can be made, you can see that now pause the video and try to write it, okay? Pause it, try it please. The conclusion for me is that the woodland with native, of course, tree species, okay, mixture of native tree species and salt cedar has higher biodiversity because the value is 0.718, the other one is 0.53 something, okay? Now that means that we have more of those rats, okay, rodents in the woodland, or the mixture woodland, okay, but they say we should explain, state and explain. Why? Why does it have higher biodiversity? Because we have more trees, okay? The trees provide more food, okay? The sources of habitat niche for this rodent to live. Okay, that's why we have higher biodiversity for it. Okay, look at it. Yeah, conclusion. The biodiversity species in the area with mixture is greater higher. Can see from the calculation. The area with mixture, okay, has more rodents. That's another meaning. But what's the explanation here? Diversity is low with salt trees because fewer sources for rodents. Okay, salt trees provide, okay, less protection. All right, shelter in the other one. Okay. All right, that's it, guys. Fig 2.1 shows a deer mouse from North America. See this? You like this mouse, right? It's cute, it's cute, but then it can be nasty sometimes. If it's in the house, running around the house can be nasty, right? It looks cute in the wild. So look at it. The biologists extended the study to investigate. So they're investigating again the abundance of deer mouse, the abundance, okay, their relative abundance, the number. The biologists wanted to test a new hypothesis that the abundance of the deer mouse is higher in areas with only short cedar trees than in trees with a mixture. That's a hypothesis. So the biologists use the trapping procedure described previously to calculate the abundance of the deer mouse in 8 areas. So did it in eight areas with only cedar trees and 8 areas with a mixture of native trees, species, so cedar trees. The biologists then calculated the mean abundance of deer mouse for each type. So 8. 8, guys. Fig 2.2 shows the biologist results. That's what we have. So this is the mean, okay, for areas with only source. This is the mean here, trace it. Okay, so and then the question says the standard error SE for the mean abundance of deer mouse. This is mouse, not mouse, in each type of woodland was 0.8 arbitrary unit. Plot standard error bars on fig. Okay, plot it. Enter the axis to plot it, guys. What you're gonna do is that there's a range we use. With that range where we have 95 confidence, okay, that range, that our calculated mean, okay, lies in the same range or is close to our actual mean. Now that's times 2 times standard error, 2 times standard error. So yeah, so that's what we're going to use. Okay, that's standard error. Now that plus or minus 2. Okay, this is minus, sorry, this is minus, not plus. 2 times standard error is what? Is 0.8, okay, arbitrary unit. So that's 0.8, okay. So bring your calculator and help me. Help me, we're doing this together, guys. But if you know, you can try it, guys. So 2 times 0.8 is what? 2 times 0.8 is 1.6. So plus or minus, this range plus or minus 1.6 is the range where we have 95% confident that our calculated mean is close to the actual mean, right? So how do I trace it here? How do I draw this? Because the question says plot it. How do I plot it here? Now if you come to each one here, check the value of one box here. The value of one box here, okay, is 10 gives us 1, 10 boxes gives us 1 unit, so 1 box will be 10, 1 divided by 10, that's 0.1, okay? So 0.1, and I want to plot 1.6. Up, there's a plus and minus, plus or minus. So plus means up it and down it. Now if I check it, all right, okay, I'm going to have from here, now from here to here is already 1, okay? From here to is 1, all right, that's 1. But how do I get 0.6? So this graph is not big enough, but I'm going to plot it anyway. So from here, okay, what you just do is just You make sure you draw this line, please remain straight line. Yeah, it should be over, above it. Okay, all right, but the graph is not enough, so I'll put a bar there where it should be more than that. Then I'll take down 1.6 downwards. Okay, so help me, let's check it. All right, you know that from here to here is 1. Okay, so yeah, so that means from here to here is 0.9, is 0.9. Okay, whatever. So here is going to be 1 point. Okay, let's do it. So here is 1. Okay, so let's count 6 boxes from here. Okay, count 1, 6 boxes, 2, 3, 4, 5, 6. Okay, that's it. Okay, so from here I'll draw. Okay, that's 1.6. Draw to reach the mean. Sorry, it should be straight. Okay, straight. So you do the same thing here. In the other bar, 1.6 should be what? So if you check 1, 1 is 10. So 1. Okay, this is from here to here. 1, 2, 3, 4, 5. I think that's 5. Yeah. 1, 2, 3, 4, 5. Yeah, 5 plus another 5. That's 1 plus another 6. That's here. Okay, that's 1 box above that. Okay, so I think I should use this. So that is here. This is the bar 1.6. Okay, so I trace it down, guys. Stay with me. Take it down. Oh, straight line. Stay straight. My drawing too. So 1.6 downwards. That's 5 from here. 5, another 5, 1. So it's going to be somewhere here. Okay, so make sure you are correct. Okay, somewhere here. So you just draw it. So 1.6 up, 1.6 down. That is why we have plus or minus. Okay, so I've drawn it. So this is the up, the lower bound. Okay, yeah, this is up for this. Now, guys, can you see that there's an overlap between these bars? Okay, there's an overlap, guys. Remember that this is not as tall as it because this graph is not enough. But there's an overlap between these. Based on what we have, if you trace the upper boundary here, upper limit, see it touches here. You trace the lower here, you can see the overlap. And anytime there's overlap, that means there is no significant difference. Okay? There's no significant difference between the mean abundance of the two populations, here and here, based on this. Okay? There's overlap between two of them. All right, let's see. Answer. Both error bars correctly plotted. We've done that. So you can see that they overlap. And of course, why do we see this is plus or minus 2 times error bar, plus or minus 1.6, 1.6 above, okay, 1.6 below. Same thing, 1.6 above, 1.6 below, but this is not enough. Okay, let's look at it. That's just 1 mark for all those work. It's too much. Explain with reference to Fig 2.1 what these standard error bars indicate about the data. All right, now I told you that standard error, all right, bars, it shows us the range. It's a calculation that shows that our sample mean or calculated mean, all right, is close to the true mean. Okay, it's close to the true mean. So we're 95% confident that our sample mean or calculated mean is close to the true mean within those ranges. Okay, and of course, from what we drew there is that there is overlap. So the both bars overlapped. That means there is no significant difference between them. Okay, we have drawn them. So because of that overlap, so how close calculated mean? How close is likely to be to true mean? Okay, there's overlap in the okay. So this indicates there's no significant difference. Once there's overlap, no significant difference. Okay. The biologists then analyze this data using a t-test compared to the abundance of the deer mouse in the two types of woodland. State a null hypothesis. So null hypothesis, all right, the abundance. Null hypothesis is always pause the video and try it. Okay, pause the video and try it. Okay, abundance of the deer mouse. Okay, so we're comparing it. So now, null hypothesis is that there's no significant difference between the abundance of deer mouse in the woodland with only salt cedars and the okay and the abundance okay there's no and that of the woodland with a mixture of trees native trees and salt cedars. Look at it, there's no difference between the abundance, there's no areas with okay it's negative apoptosis. Oh, that's another question. Mosel mantis edelis are moths that live in seawater on the shorelines of coastal regions around the world. Mosel are popular seafood and so are widely cultivated. After harvesting the mussel farmer must replace them with young mussel. This is often done by collecting young mussels from the wild marine ecosystem. So read the question. Fig 3.1 shows mature mussel after harvesting. That's cute, man. How does it taste? Okay. The sleeper limpet Credipula fornicata is an invasive alien species introduced into Europe from North America. Sleeper limpets compete with mussels and reduce the yield of mussels. Sometimes sleeper limpets accidentally collected with young mussels. Okay, mussel farmers want to prevent the introduction of sleeper limpet when restocking, okay, their farm. So read this again and again. Fig 3.2 shows the sleeper, the limpet. See the limpet. How does it look? Okay, let's continue. Scientists investigated the best way to kill silver limpet because it's an alien species. They want to remove it without harming the mussel. This is what we want. We want to take out this guy. The scientists put 30 slipper limpet length greater than 30 millimeters into each of the four trays. Each tray of slipper limpet was exposed to a different test condition for a period of 3 days at temperature of 12 degrees Celsius to 13 degrees Celsius. The four test conditions are shown in table 3.1. Now I can see that. See the different conditions: air exposure, brine rains, okay, and repeat brine rains, brine soak. This is what I mean. The fungus is exposed to the air. Okay, read it at start. The one this is soaked. These are different conditions. Okay, live organisms exposed to the air at the start of day one, raise organisms with saturated salt, that's sodium chloride, that's brine. Saturated sodium chloride is brine. Okay, for 5 minutes and then expose them and start day one, rinse with saturation or solution for 5 minutes and then, okay, these are different treatments, right? Let's go on. This procedure was repeated with one tray of 30 slipper limpet. Okay, there's one tray less than 30 millimeters, and then one tray 30 millimeters greater than, okay, for each of the four test conditions. After 3 days, the scientists counted the number of dead slipper limpet and dead mussel in each tray. You see a lot of information here, but it's okay. Absorb it. That's how it rose. The scientists repeated the whole investigation at a temperature of 4 degrees Celsius to 5 degrees Celsius using fresh samples of slipper limpet and mussels. The results of these tests are shown in 3.3a. Okay, so look at it, guys. A young Muslim farmer concluded from this is what this guy, a young Muslim farmer observing a scientist do this, concluded from the result of the investigation shown in Fig 3.1, 3.3 that young mussel used to restock the mussel farm should be, they said they should be given a brine soap treatment to kill any sleeper limpet that are evaluate this conclusion. So I want us to evaluate it. So you should use the data in Fig 3.A and Fig 3.3b to support your answer. So evaluate means you should assess it. Okay, make argument for and argument against. Okay, logical. So it's saying that they should stop using brine soak treatment, which is the fourth one. Is brine soaked. That it kills kill any sleeper. So is it correct? Does the data support what it's saying? Okay, let's look at the data. Let's see whether there's something that supports it. Yeah, if you look at brine soak, okay, for 4 degrees to 5 degrees, okay. All right, if you look at it, you see that brine soak killed all the limpets. Okay, can you see killed all of them? This is sleep. The black one is sleep limpets greater than 30, and this blue one ash less than. So it killed them at this temperature, all of them. And here it's also killed them. So brine soak treatment kills, all right, eliminate 100%, okay, of the sleepet limpet. Mhm. Both in both conditions, 4 degrees Celsius to 5 and then 12 degrees to 13. That's what it showed here. And of course, if you check, you see that for 4 degrees to 5 degrees, this is another point, right? We have fewer, um, fewer mussel killed. Remember, we want to preserve mussel, but fewer of them are killed in this temperature compared to this. But some of them are killed here too, but fewer. Okay, so this supports it, right? But also you can look at it, other treatment. Let's look at other treatment. Remember that, look at it, this is treatment, 3 repeat prime kills 100% here at this temperature. Okay, with fewer muzzle, okay, killed. So it should be the best because the soak one, prime soak, this temperature and that kills more, right, of muzzle, but this kills less, fewer. And also if you check it here, all right, for this temperature it kills none. Can you see no muzzle killed using 3 repeat primaries at 4 degrees Celsius to 3 degrees? So this could be the best because, okay, our muzzle is not being killed. Okay, all right. Now, but you see, you can also say that we need to do a statistical analysis, do a statistical test for this. Okay, all right, because this is not enough. We need to do more replicates of this and use statistic test on it. Okay, and do more number. Okay, let's do what it said. Any for from branch soak treatment for kills 100%. You see, everything was said here. Okay, that's it there. Okay, tested 30, not enough. Okay, that is it. If anytime you're evaluating, you're assessing, making argument for, all right, arguments. Okay, conclusion not tested. Okay, yeah, 4 marks. So thank you for staying with us. See you in series 5. This is Shedro. Bye.